Okay, so this is a splicing branch point. This has a pretty unique structure, as you can kind of see here. And so again, that's that trans-esterification reaction. It's going to generate the branching point. It will form that lariat loop intermediate. And that's what you're seeing here. Normally this would be like this, right? So 5 prime, 3 prime sort of stuff. Da, 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 da. And instead, now we have this 2 prime as well, don't we? And can you see that nice loop intermediate, lariat intermediate? Why do you never see this stuff on DNA, by the way? No, it's okay. So here, this is that branch point, right? And this is going to form the head of the lariat, right? Where it comes together. Okay. Now, why do you never see this stuff on DNA? So what is the normal direction this is supposed to go? Which of these is unusual? So you're supposed to have the 5 prime, you're supposed to have the 3 prime, right? Okay. Which one's the weird one? 2 prime. What does the D in DNA stand for? Deoxy. So can DNA do this? No. So this is why you never see this in DNA, right? You can't actually form a lariat in DNA. Okay? Okay. And so ta-da, we have our beautiful lariat. Everybody appreciate our lariat? Fantastic. Yes, it's beautiful, gorgeous. And what points do these typically form at? What's the base that these normally form at? Yeah, A. That's the base right there, and G. Got it. Okay, so the small nuclear RNAs and spliceosomes can catalyze the splicing of the mRNA precursors, the small nuclear ribonucleic protein particles, SN, small nuclear, RNPs, ribonucleic, and then particles, P, and the S is because we have more than one. Small, they're going to be a less than 300 nucleotides. Remember, in cases of DNA, RNA things, less than 300 is very small. Okay? We're usually talking like thousands, tens of thousands, right? And so the RNAs that facilitate splicing, these are U1, U2, U4, U5, U6. These are essential for the splicing. The secondary structures are going to be highly conserved. They're associated with these specific proteins that will form the small nuclear ribonucleic protein, or SNRNP again. Yes, this was not particularly well advised. Particles, protein, both having the same abbreviation, right? Yeah. So the SNRNPs is going to be associated with hundreds of splicing factors and the mRNA precursors to form the 60S spliceosomes. So right here we have the U1, U2, U through U6. These are the size of the SNRNA. And again, those will be the nucleotides. These will be the small fragments that we have. These will be the specific roles. So for example, the U1 is going to bind the 5' splice site. The U2 will bind the branch site. The U5 is going to bind the 5' splice site. And then the 3' splice site. The U4 is going to mask the activity of your U6. Then U6 is going to catalyze splicing. And so right here we can actually see this assembled together. And so we have the spliceosome. Again, this is going to have a protein and RNA components. And here we see protein component. And over here, these colorful portions, we see RNA components. Everybody okay with that? Okay. Good God, you almost certainly can't see that, can you? Okay. I really need to stop doing these on my computer at home. The screen is way bigger. Okay, and I need this part. Mm-hmm. So I don't do it by number of questions, actually. I do it by the time it would take you to do the final. And what I do is the 25% slowest have to be able to complete it within that amount of time, which will be 2 hours for the final. And so it will definitely not have the same number of questions. None of the tests will actually have the same number of questions for sure, but the final will be two 2-hour time periods. And so it'll be like taking 4 of the normal exams, okay? In terms of time it would take you, and the questions will be proportional to that, but again, it's not exact, because, yeah. I mean, I did give you some hard questions as well, but yeah, you should expect to be able to spit the easy stuff out pretty quick. I'm giving you 1.5 minutes to do each one, even though they actually should only take about 30 seconds for me. Yeah. So, yeah, the finals will take between 1 to 2 hours, depending on how fast you are. Did that answer the question? Yes, we'll have 2 2-hour periods that you'll need to take it over. I guess theoretically, if you're fast enough, you could probably do it in 1. And I could do that, actually. That would be fine. That totally sounds like something I would do. All right, so anyways, here again, we're going to have that spliceosome. It's going to promote the splicing of some of the genes. So here we're going to have the U1 and U2. These are going to bind the 5' splice site here, and the U2 is going to bind at the branching point. And we can actually see that right here. Again, this is the 5' splice site. Notice that's that G right there. Right here, that branching point, that's that A. Everybody see that okay? And so remember that these are not, it's not magic, right? It's not like it has a consciousness guiding it. This is not something that can just go, oh, we have the branch point here, we need to line these up and use your hands, right? This is something that needs to have tags that are going to identify each portion of this, right? And so this is essentially your tagging system. Hey, this is where you're going to cut. Hey, this is where you're going to join. Now these are identifiable, okay? And so we've basically tagged these. Okay, notice that this is going to be at the end of the exon. This one is going to be right before the beginning of the next exon. Everybody okay with that so far? Fantastic. Okay, so what's going to happen next is you're going to have the U4, U5, U6 complex join onto here. And this will be preformed. It's going to come on as a complex, and then it will form an overall complex with the already attached U1 and U2. Now notice that when it forms this overall complex, this intron, does the introns always the same lengths? Dang. I thought I wouldn't have much attendance today, but it's doing really good. We're actually doing great. All right, we're doing awesome. Does the intron always need to be the same length? No, it can be quite variable, right? And so this is going to be where the complex comes on. Your U1 is going to join. Your RNA will bend. It's that single strand. And so it'll bend over. And then you'll have your U2 join. See that okay? And then this exact length of this loop doesn't really matter that much, does it? It just needs to be long enough that it could bend all the way back in and not, hit the complex. Okay? Okay. And so what's going to happen next is your U1 and U4 are going to come off here. And let me see, where am I? So again, you're going to then get the U1 and U4 coming off, and this is the one that's going to bridge over here, by the way. Can everybody see that? Okay. That's the one that matches here. Okay. Yeah, I had to cut it up. This is also where you're going to have the U6 snRNA. It's going to refold and then bind the 5' splice site. It'll displace the U1 actually. Notice the U2 actually stays attached. This U2 is actually going to be what displaces the U4. So in this previous complex, can you see that it was bound right next to the U4 here? So what you're going to get here is the masking of the U6, which is the U4, is going to come off. This is going to have the U2 move up onto that U6, and then the U1 is going to pop off, allowing you access to that site. Everybody see that okay? Fantastic. Okay, so this is going to be where you can now perform the first transesterification, okay? You can do the first transfer. And again, the U6 is unmasked, and the U2 has moved up into the site. So you'll actually notice this arrow here that's telling you you're doing your first transesterification right there. And so that's the reaction that does that. And then down here, we're going to have the second transesterification right there. So the first transesterification step, your branch site, the adenosine, is going to attack your 5' splice site. This will make your lariat intermediate, wrong one, right up here, right there. And so we'll have our lariat intermediate right here. Everybody say okay? Okay. Notice what is still on there. So we've split one side off of our intron, right? This one right here. This one here, though, is still on there, isn't it? And so you just have your lariat intermediate with your exon still attached on that 3' side. Okay, and so what's going to happen then is you're going to have this second attack. This is the second transesterification. That's going to join your exons together, and that will give you your spliced exons. They'll drift off. Okay, and then that will end up giving you your lariat intron right here. And again, that's the just the intron portion. Anybody have any questions about that? No? Okay. So at that point, we actually had the exons linked together, and we have excluded the intron completely. Okay, so again, these are the steps in that overall process. We have step 1, which is the U1 snRNP. This is going to contain a conserved 6-nucleotide sequence. It's going to bind to the pre-mRNA 5' splice site. And again, this is a highly conserved sequence because it actually has to bind on to this sequence right here, which is the one that says, hey, we have something that needs to get spliced off, right? And remember that it needs to do this in such a way that it's going to bind right here, which is right after the upstream exon. Okay? If you mess this up, you would actually start cutting off the exon. Or you would start leaving groups on in the mRNA into the exon. Okay? So it has to be very highly conserved. Step 2, the U2 snRNP. This is going to bind the intron branch site. This will require the ATP hydrolysis. So step 1, U1 bind. Step 2, we're going to bind the intron branch site. That's the one downstream. That's the one down here. Remember that one over there, right there? Okay. Okay, now at this point, we have these two sides labeled. So step 3 is going to be recruiting, bless you, the U4, U5, U6 tri-SNRP complex. So this is going to bind the complex of the U1, U2 to this U4, U5, U6 already assembled complex. Okay? And the mRNA precursor, this is going to form your spliceosome when all of these are together. Remember that the U1 and U2 will already be bound to the mRNA precursor. This will then bind that U4, U5, U6 tri-SNRNP complex, and then it binds it on the other side, and that gives you your full spliceosome. okay, ready to act. Again, this is going to require ATP hydrolysis. This is a non-spontaneous process. So the spliceosome calyx center, this is going to have 2 key magnesium ions. You guys probably already kind of knew that somewhere in the back of your head, because when we were doing things with ATP hydrolysis, what did they almost always have? Actually, I'm pretty sure they always have it, but I'm hedging my words. You guys can discover one that doesn't, and then you can come back and go, Dr. Clausen, liar. And actually, I would like that. I would like to know. Go find me some ones that don't have magnesium, okay? Okay. So anyways, you probably already kind of knew that, because usually when you're binding anything with phosphate groups, what do you have? Magnesium, right? And so these are the 2 magnesiums. They're going to bind primarily the phosphate groups from the U6 on RNA right there. And remember, the U6 is the one that actually does the snipping, right? Okay. And that's the one that's masked by U4. So step 4, this is rearrangement in the spliceosome. This is going to then occur. Your U1 and U4 is going to be displaced from the spliceosome. U6 is going to undergo an intramolecular rearrangement. This will permit the base pairing with the U2 as well as with interaction with the 5' of the intron. And so U4, your masker, U1 was in that first site at the beginning, upstream, right? And so those are now gone. This is going to allow those 2 groups to come together in the U6, and now you're on mass so it can do its catalytic activity. And that involves a rearrangement, right? Which, not surprising, probably. Step 5, the first trans-esterification is then going to happen because we're catalytically ready to activate, right? We have the groups near each other, we're catalytically active, we're going to go. And so, first esterification, that's going to be the cleavage of the 5' exon, and this is going to give you your lariat intermediate. And that's actually where we're seeing right over here. This is the reaction. This is the result of that, okay? And so we snip off and we move it to the branch point. Remember, the branch point is downstream, okay? This is one of the more common mistakes people make, by the way. Step 6 is going to be the U5. This is then going to align the free 5' exon with the 3' exon, and the second trans-esterification is going to occur. So step 6, the second trans-esterification, we move the exon group off onto the other exon group so that we have those joined and ready to go. Okay? This will end up snipping out the entire intron now. So this actually results in your mature spliced RNA and the U2, U5, and U6, which is actually bound to the lariat intron. And so this actually drifts off here as a complex. Okay? And this can then go do other stuff. But this actually drifts off the complex, and you're left with the exon by themselves. Okay? Oh, you have to go, ooh, ah, so beautiful. It's like a ballet. Okay, so the mutations in either the pre-mRNA, this is the cis-acting, or the splicing factors, which would be the trans-acting. This can result in pathological conditions like we had kind of hinted at before, where a little bit of mismatching can cause vastly different behaviors, right? So defects in splicing are going to be estimated to cause about at least 15% of all genetic diseases, more than you probably thought, wasn't it? Yeah, you all thought it was probably DNA, didn't you? Blaming everything on DNA. Poor DNA. How could you do this to it? Okay, sorry. I'll stop trying to make jokes. So some thalassemias, these are diseases that result from defective hemoglobin synthesis, either caused by mutations at the 5' or 3' splice sites in either of the 2 introns of the hemoglobin beta chain or in its exons. Actually, one thing I would like to know, by the way, do you guys find this helpful where I actually use real diseases to talk about it? Okay, or is it a waste of time? Okay, so retinitis pigmentosa, this is a disease of acquired blindness. This is due to the mutation in the pre-mRNA splicing factor. This is a component of the U4, U5, U6 try SNRNP. You may notice, by the way, this is probably my weakest thing because this is the only time I'll ever get to say these diseases at all. And in fact, even if I was like a doctor around here, most of these diseases I would never get to say in my entire lifetime of treatment, right? Anyways. So mutations in the splice sites, these can be caused by diseases such as thalassemia, of course. Here we have a normal pre-mRNA. This would be the in-frame stop codon right here. And so what we see is that the pre-mRNA with the mutation in the intron, instead of the AU here, like we should have, we have GU instead. And so this actually gives us a different new 5' splice site. And so instead of your normal mature mRNA, which would have had these right here spliced out into the exons, and then these are the introns, right? Then instead we get this. And so we have spliced in a new stop signal. So it goes, protein, making protein, and stop. And we skip 2 of the exons. Whoops. That's kind of a half-assed job, isn't it? Whoopsie. And by the way, is it actually such a big deal to get the wrong amino acids in frame? Like if you added an extra amino acid in there, is it always going to cause problems? There's actually a pretty high chance it won't. So like if you're in a polar region and it just, is a loop that makes a polar region and interacts with the solution, it don't really matter if you have 5 or 6 or 7 of them. It just loops back, right? No biggie. If you're in a non-polar region and it doesn't really affect the binding site, it's just structural region, it doesn't really matter if you add another non-polar or neutral one. There's a lot of actually non-conserved regions that doesn't really matter. The only time that really matters is where it ends up in a conserved region, right? And that's why we had talked about that mutation effects in that previous chapter, which is actually really interesting material if you're going to go into that stuff. Okay? That basis will take you really far in that field, by the way. Anyways, a lot of you probably won't go into that field, which is fine. But it's things like this that really kill you. It's where you get things like a stop signal, because then you end up missing like 2/3 of your protein, right? Or half your protein or something. And you're pretty likely to end up cutting something important, right? Yeah. This is where it really gets damaging. So most human pre-mRNAs can be spliced in alternative ways to yield different proteins. This stuff starts getting really complex, and we're not going to be able to get that far into it. Not that we would have been able to get that far anyways. This is a field that is just blossomed over the last 20 years or so. And God, you could spend a degree, a PhD on this alone. So anyways, alternative splicing. This is going to be a mechanism by which the different combinations of the exons from the same gene may be spliced into mature RNAs, which will then make different proteins and express different ways in your cells. And so the mechanism for generating protein diversity, it's going to lead to changes in your coding sequences. The splice sites selection is going to be determined by your binding of your trans-acting splicing factors to your pre-mRNA cis-acting sequences. Does everybody understand trans-acting and cis-acting to some degree? What does cis mean? Mm-hmm. You got it. Same. Trans means different. Yeah, and so different acting changes how you splice, right? Cis acting, same. Doesn't change how you splice. Okay, so here we have alternative splicings. They're going to produce the different mRNAs. We have our pre-mRNA right here. And so in neuronal cells, because of how it binds to this RNA, what you have binding to it, you'll have different splice sites that are available to bind to, right? And so what you'll end up getting is different splicing in the neuronal cells versus the thyroid cells. And so you end up with CGRP in the neuronal cells. In the thyroid cells, you end up with calcitronin. And that's from the exact same pre-mRNA. Okay? And you can actually see some parts are actually reused. See these? These are all reused. Okay? It's just this portion gets spliced out, ends up being an exon here, and this portion, which is blue here, ends up being the exon over here. In the other case, this would be an intron, right? So here's some selected human disorders that are going to be attributed to defects in alternative splicing. I don't think you guys need me to necessarily read these off for you, right? Though I will note there's some really interesting ones like hemophilia A. Some pretty cool stuff, actually. Well, not cool for the people that have it, of course, but interesting research subjects. Transcription and mRNA, this is going to be the processing are going to be coordinated by the CTD of the RNA polymerase II. The CTD is going to consist of the repeated sequences YSP, TSP, S. So the S2 and S5 are both going to be phosphorylated. The phosphorylation state is going to direct the functions. The CTD is going to contribute to the efficient transcription by the recruiting proteins to the pre-RNA, mRNA. This will include things like capping enzymes, components of the splicing machinery, endonuclease that will cleave the transcript at the poly A addition site. And here we have a sort of example. So this is again the CTD. It's going to couple with your transcription to your pre-mRNA processing. Remember, here would be your transcription, right? You'd be normally coming off this as you're going along. You're going to be feeding this out. Can everybody see that okay? Okay. Okay, so this is your head right here, and so this is going to be the phosphorylated CTD. This will be your capping enzymes here, splicing factors here, polyadenylation factors here. And so what you're going to see is that it's going to be coupled to this. So you come off here, the first thing that hits this is going to get capped, right? Because that's your feeder. That's what you're going to feed into your ribosome eventually, right? And so you put a cap on there so you can feed it in, okay? The next thing that comes along is going to be involved in recruiting spliceosomes, right? And that's where you'll start splicing out your introns so you can get your exons grouped together. Now remember that you might have other stuff which will bind to different parts of this depending on the cell and obscure those branching points, right? So that you can't do it at certain sites. Okay, and then the last thing that's going to happen is as you come along here and you end this, you're going to end up with your poly A tail attached to it, right? And so this is going to be the portion that's going to add your poly A tail. Ooh, ah. So wonderful. Okay, did everybody catch that okay? All right, so your small regulatory RNAs is going to be cleaved from the larger precursors. Your microRNAs is going to be small 20 to 23 nucleotide RNAs that will regulate your gene expression in your eukaryotes. They're generated from the initial transcripts produced by the RNA, polymerase II, and sometimes RNA polymerase III. They're going to fold into hairpin structures. These are going to be cleaved by specific nucleases. This hairpin structure, you're going to see this a lot in like bacteria and stuff because that's what it mostly uses for signaling. But essentially think of these as the bacteria signaling, but in a step before so we can then use it as the tags. Does that make sense? Yeah. So again, we still see that hairpin motif here. We're just adding a step in, right? Making it a bit more complex, a bit more regulated, okay? Or nuanced would be a good way of saying that. So again, we'll have that folded hairpin structure. We'll have that cleaved by specific nucleases. It's going to be bound by regulatory proteins in their single-strand form. This is then going to help target the gene regulation. This is what that looks like right here. So we have the initial transcript. Why do we get the hairpin turns, by the way? It's just bases matching, right? So your base sequences match each other. That's all that is, okay? Remember, they're just going to be drifting and coming off and on, and if they match really well, they're going to bind, right? And that'll make a hairpin turn, and then anything that's going along here, like those complexes we've been talking about that are feeding along it, they're going to hit that and go, well, oh shit, this is what happens, okay? Okay, so again, we have our prime site. We have our poly A tail here. We're going to get cleavage. This is going to cleave off the 5' cap and poly A tail. Why do we cleave those off? What the hell, Dr. Clausen? Did you talk about those being necessary for a ribosome? Are these going to make the protein? Oh, they're going to be bounding the protein, right? So you don't need the poly A tail. You don't need the 5' cap on these. Okay? This is then going to have cleavage. This is going to cut these into 2 mature microRNAs. So your microRNAs then are right here, again, 20 to 23 long. They're going to bind a regulatory protein. And now notice what you have. What was that like? Basically, you just took Velcro, stripped it apart, right? Cut off a length. And now you have something that can stick on to the portions you want to regulate, right? There you go. Yeah. Kind of cool, huh? Oh, you're too late in the semester, aren't you? You're like, to the point where I just don't care anymore, Dr. Clausen. Just leave me alone. All right, so the RNA editing, this can lead to your specific changes in your mRNA. The RNA editing, again, is going to be the post-transitional, transcriptional, change in the nucleotide sequence of the RNA. Some of the examples will be the codon for an amino acid. This may be changed to that of another amino acid. A codon for an amino acid may be altered to a stop codon. The RNA editing is going to play a role in these processes, which is the process of lipid transport by the ApoB, the regulation of the postsynaptic receptors, changing mitochondrial RNAs, mRNAs, into trypanosomes. These are actually parasitic protozoans. So right here we have the RNA editing after your coding sequence of the mRNAs. Everybody can see that figure okay, right? Looks pretty decent. Okay, so your enzyme is going to catalyze deamination of the specific cytidine residue in your mRNA for the apolipoprotein B100. This will change a codon from the glutamine CAA to a stop codon UAA. So here we have the lipoprotein assembly. Here we have the LDL receptor binding. So the ApoB100, if we had the unedited mRNA, this would have translation, which would go to the ApoB100. This is then the deamination. Instead of having CAA here, we have the deamination occur. So we go lose an NH4 plus, again, that being deamination, right, as in organic chemistry. And this was going to give you the UAA. That's that difference between C and U. This is then going to be a stop. And so instead of translating all the way through, we would actually get a stop here, which would be this blue portion right here. And that's the ApoB48. By the way, what do you think these numbers are? 48 versus 100? That looks suspiciously like 48 out of 100, doesn't it? Yeah, that's what that is. Okay, so the ribosomes is going to be RNAs that function as catalysts. Your group 1 self-splicing introns are going to be the introns that excise themselves. These will be initially identified in the rRNA. This is from tetrahymena, This requires guanosine as a cofactor. And we can actually see this under a microscope here, which is quite interesting. Okay, so some of the introns are going to be capable of self-splicing. We don't need that entire spliceosome complex to do it, okay? And this is actually, so there's a thought process that these are actually where all of this mechanism came from originally. that the self-splicing ones originally were what were developed, and then the spliceosomes developed beyond that. Okay? And so right here we have the upstream exon. Here we have the downstream exon. Again, this is an intron right here. And note that if you get a portion of this that tends to fold together and come in close contact, because we have that diol. In organic chemistry, remember diols were quite reactive, right? And so if you bring those into close proximity to each other, there is always a chance that you will get esterification. And so what you see here is you get the upstream exon. It's going to attack that downstream exon. And this is going to cause that spliced exons. Now, this would then leave this free intron. The free intron is then going to have these two ends, which will be reactive, and so they'll stick together. Now, note that this tends to cut off a little portion of it, a little lariat, okay? Except that that lariat, it's not very stable, and so it loses a fragment, whereas the circular DNA is more stable, okay? And so this actually is kind of interesting because this circular form can actually open and close and open and close. And you can have little fragments that get cut off depending on their stability. Okay? And this would actually happen naturally if you were to have a group like an RNA in solution and it has different stabilities at different one of these bases, right? So you'd actually get naturally cutting it off. Like this. And so we actually can see this goes from 399 to 395 actually, and this is actually known as the L19 RNA right here. And you can actually see this in action, which is pretty cool. Okay, so this is the self-splicing introns. These are going to have very complex structures. Here we have the guanine binding site right there. So the catalytic mechanism for your group 1 self-splicing intron is going to include the series of trans-esterification reactions. Your splicing sites are going to be aligned with the catalytic residues by base pairing between the internal guide sequence, IGS, in the intron, and the 5' and 3' exons. Please note that I know the figure here makes it look really, really simple, like this doesn't have any complex geometry going on, but them being put into an angle that they're reacting easily very much means there's complex geometry going on. It's binding to itself. It's doing things like that, right? And this is where you're starting to see that. And then we see that in more detail right here. Why are these things coming in contact and having reactions occur? Because they have sequences of bases that match up, right? That's what's stabilizing that so it's in the correct orientation to react, right? And making those groups more reactive. So again, right here we have the internal guide sequences. These are, can you guys even see that figure? Yeah, again, right? Shit. Yeah. No more doing this on my home computer. Okay. Can we see it now? Yeah, part of the problem is this projector is just not bright enough for this room, right? Yeah. Which, whatever. Okay, so right here we actually have the upstream exon right here. Again, a phosphodiester bond will be formed between the two exons, and the intron is going to be released as a linear molecule, which can then fold back into on itself in a loop form. So this will be the first part right here. Here we have everything joined together. We have the upstream exon. Right here we're going to have the IGS. It's going to fold back onto itself and match. That's why it's the guiding sequence, right? Is because it's the one that will have enough matches that it will be stable in this form and cause this to wrap back out around on itself. And so this is then going to be the intron that will happen after the IGS here. This then is the downstream exon. And this right here, these P's, these are the phosphates that are going to be involved in that phosphodiester bonds that are going to be the ones that will be reactive. And so, again, note you have a fairly complex geometry that develops here, okay? And you can see an example of how complex this can look right here, okay? Again, please note that your A's and G's tend to be the ones that are the more reactive, less stable ones that tend to do these reactions, right? Okay, and so what we're going to see is, again, we get a G and A, don't we? And we're going to get that phosphodiester doing that transesterification. And so you get this A here. It's going to react with this OH here, again, on a guanine. And this is going to attack that phosphodiester linkage right there. Note that A, again, frees up that 5' end first. Everybody see that? And so what's then going to happen is that 5' end is in close proximity to the 3' end one. And that's going to, again, get that second transesterification that attacks and reacts. The big difference here being that we don't need a spliceosome and all that mechanism, right? We have little segments of the DNA that are doing the moving them into a reactive site themselves. Or RNA, excuse me. I mean, the segments of the DNA would also correspond, right? But yeah. And so then we have our spliced RNA here. The exons will be joined, and we'll free off. And again, this is that AG and G right here. And again, this would be a linear intron, and it can then wrap back around on itself to form that circular one that we saw later. So in the cell splicing mechanism, the calyx site will be formed by the intron itself. In the group 2 cell splicing introns, the attacking group is actually the 2' OH. This is on the adenylate in the intron, which is that A attacking that G that we were looking at just a bit ago. Okay, and so again, this is group 1 here. This is group 2. This is the spliceosome. And here we can compare these different ones. And can you guys see those figures well enough to compare them? Fantastic. Okay, and so these ones are both self-splicing. So again, we have group 1 and group 2. Now, there's the big difference between these is that in group 1, we actually have the G here that's going to attack from that 3' position onto that phosphorus right here, that phosphate. And then the group 2, instead of it being a G here, we actually get the A here attacking, and that's in the 2' position. Now, you'll notice that group 2 looks a lot more like the spliceosome, right? Which also has a 2' A that attacks. Okay? So the next step, group 1 here, then has that 3' attacks that 5' end of the exon. Again, that's the exact same for group 2. And then we look at the spliceosome, and it's the same, isn't it? Yeah, they all look the same. Then over here, we get the group 1s. Hey, what's going on here? Is that lariat-like? Is it coming back on itself? Well, we get linear, don't we? Yeah, so in the group 1, the biggest difference that you'll notice is that we end with linear, actually. We don't end with a circular one. Okay? Group 2, on the other hand, ends with a lariat, and the spliceosome ends with a lariat. The group 2 and the spliceosome versions, these are very, 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 very similar. Okay? They really don't have much of a difference at all. The big difference being that it's an internal match in group 2 and the spliceosome and the spliceosome 1. Okay? Which is matching to an external RNA that's attached to the spliceosome, right? Okay. Everybody see that okay? Group 1, on the other hand, does not. It's a linear that you end up with. Now, it can later on reattach into a circular form, but you don't end with a circular form. The RNA molecules can also play a role in the protein biosynthesis, the catalysis of the peptide bonds, the polymerization of the other RNA molecules, or even of themselves. In the vitro experiments, these have been identified, the possibility of a self-ligating ribosomes, the RNA molecules that are capable of joining other short RNAs to their own end. In vitro experiments have identified these possibilities. Keep in mind that even though there's so much complexity that we've already started building out and fleshing out, we still don't know everything. Okay? This is a continuously evolving field. Really, the amount of literature in this field has about doubled every 40-something to 60 days for the last, 10 years. Okay? So it's a massive undertaking. Okay. So there are parts that we still don't fully understand. Anybody have any questions? I guess I'm probably just going to end here today, since starting a new chapter with 3 minutes left seems like it's not particularly useful. And we'll start